All right, well, that first recording wasn't as short as I had hoped, but it was about a half an hour. I guess that's not too bad. And that was chapter 5.1, Modifications of Mendelian Ratios Involving One Gene. This is chapter 5.2, Modifications of Mendelian Ratios Involving Two Genes. So first thing, what does a Mendelian ratio look like involving two genes? And that means we're modifying a dihybrid cross. So the simple representation of a dihybrid cross is this. Now, a dihybrid means two genes, right? So dihybrid cross is a cross involving two genes. We take something that's homozygous for these two genes and homozygous for the recessive genes. This is one way to do it. And if you do that, you generate a dihybrid. Cross two dihybrids together, that's a dihybrid cross, and you get this pattern. It's all about patterns. If you see a 1 to 2 to 1 or a 3 to 1 or a modified version of 1 to 2 to 1, that's one gene in a monohybrid cross. A dihybrid cross always gives you something that looks sort of like this. And what we know, if we see a 9 to 3 to 3 to 1, or as we'll see in this chapter, modified versions of 9 to 3 to 3 to 1, that means it's a dihybrid cross. That's a very specific thing. If we see 9 to 3 to 3 to 1, we know this. And that's the key to this whole chapter. Is if you see 9 to 3 to 3 to 1, 9 to 3 to 4, 9 to 7, 12 to 3 to 1, which I'll show you a table of all those things in a minute. All of those we can interpret as modified versions of 9 to 3 to 3 to 1. And this is where we have to use this abbreviated genotype thing. So this is it. This 9 to 3 to 3 to 1 with these symbols is how you interpret a dihybrid cross. We'll never have to add all the different types of genotypes. Just use this abbreviated version. Now, one thing I'll add is that you can get to this dihybrid situation like this too. So this one's homozygous recessive for the A gene, homozygous dominant for the B gene. This one's homozygous for the A gene, homozygous dominant for A, and homozygous recessive for B. That'll still give you this. And anytime you get to this, which is a dihybrid cross, and that's the only thing that is a dihybrid cross, then you get this. So just get very used to doing this. You don't have to do any Punnett squares. You don't have to do anything. You just write this down. So at least you don't have to memorize it, right? Because it's an online class. All right, so that's basically it. Let's throw in the middle here. We'll do a new page. But also remember that there's a test cross. So a test cross involving two genes. A dihybrid test cross where it looks like this. A dihybrid, heterozygous for two genes, and a test cross. And we call this the tester. That always, until I tell you otherwise. So what I didn't mention or remind you of before is that 9 to 3 to 3 to 1 tells us two things. There's a dihybrid cross going on, and the genes are assorting independently. So up until now, and this whole chapter, all the genes are assorting independently. Chapter 7 is where we look at genes that are not assorting independently. But right now, genes are assorting independently. So if genes are assorting independently, that means we get this kind of, we always get 1 to 1 to 1 to 1. Now, remember, so our 9 to 3 to 3 to 1, which is a dihybrid cross, we put in these little dashes to say that we can have either a big B or a little b or a big A or a little a in each of these cases. But in these cases, in a test cross, we know what the second allele is. So if this is, so the reason that we get this is because this guy gives us these types of gametes, big A and big B. So big A and big B can go together. Big A and little b can go together. Little a and big B can go together. And little a and little b can go together. And these are the gametes. So gametes are really important. Then the tester only gives us little a or little b. And so for this individual, we can get big A and big B from the parent on the left here. But we always get little a and little b from the parent on the top. In the next one, we can get a big A and a little b from the parent on the left, but we always get a little a and a little b from the parent on the top. The next one, we can get a little a and a big B from the parent on the left, but we always get a little a and a little b from the other one. And then the bottom one, we get little a and little b from the guy on the left, and little a and little b from the guy on the right. So that's always, always, always what we get from a test cross when the genes assort independently. Now, the cases that we've seen for a dihybrid cross where we get 9 to 3 to 3 to 1, that's because we've been looking at different traits. So we might have tall and green, tall and yellow, short and green, short and yellow. So we're looking at two distinct traits, height and color. What this chapter is all about is now we're going to look at a single trait, like color, but it's going to be controlled by more than one gene. And that's a really important concept in genetics. It's really rare, and it's really never true at all, that one gene is the only thing that is involved in a particular phenotype. There's always multiple genes. And so the way to enter that sort of concept is to start with two genes. All right, so the good news is that everything has this pattern. And all we're going to do is we're going to see modifications of a 9 to 3 to 3 to 1 ratio. And what those modifications are going to be are where we combine different versions or different categories. So sometimes these guys will all give us the same phenotype. And so we'll get 12 instead of 9 and 3. And then we'll get 3 of these and 1 of these. Sometimes these guys will get combined, and that will give us the same phenotype. And so that in that case, we would get 9, 3, and 4. Sometimes this gives us one thing, and all of these guys give us something else. So we'll get 9 to 7. And so there's a table in the book, and I'll show you that table in a minute after I go through some examples, but there's a table that summarizes all the different combinations we can get. All right, so let's just start with, and we'll just go through the book's order of things, and go from there. All right, so the first example is going to be a having to do with some kind of peppers. All right, so let's look at some peppers. So we got some peppers, and we got red peppers, and we got cream-colored peppers. And these are always, everything that we do here, we start out with 2 true breeding parents. So we know that they're going to be homozygous for both genes. But homozygous means they could be homozygous dominant. So true breeding means that they could be this, they could be this, they could be this, and they could be this. So it just kind of depends on the cross. Now, normally, most of the time, it's going to be these two guys crossed together, but sometimes it might not be. But in the middle, we're always going to get a dihybrid in the F1. All right, so how do we know that? It's important that these first ones are true breeding. We don't want you to have to try to guess what the genotype of the parents are. We just know that they're true breeding. And then we get in the first generation here, we get all red. So at this point, we don't know what's going on, right? So it could just be one gene involved, and red could be dominant to cream. That would make sense. If that were the case, then this would be a monohybrid cross. If it's a monohybrid cross, we would expect 1 to 2 to 1 genotypic ratio and 3 to 1 phenotypic ratio. But we don't see that. In this case, what we see is a 9 to 3 to 3 to 1. And so that tells you that this is a dihybrid cross. And what the 9 is in this case is red. The 1 is cream. So that's what we started with. And 3 are peach. And 3 are orange. So we're talking about the color of the peppers. And so that's the trait. So there's one trait, but it's controlled by 2 different genes. We know that because we get a 9 to 3 to 3 to 1 ratio. And a 9 to 3 to 3 to 1 ratio, by definition, is a dihybrid cross. So we can use whatever letters we want. For some reason, I guess the real letters for these genes are Y and C. So if you look at this example in the book, you'll see Y and C, but we're just going to use A's and B's just to keep it consistent. So we know that every time we get to a dihybrid cross, we get this. Every single time. If the genes are assorting independently, which they are in all of these examples, until chapter 7. Then we won't do dihybrid crosses anymore. So we get this, because that's 100% of the time what you get when you do a dihybrid cross. We know it's a dihybrid cross because we got a 9 to 3 to 3 to 1 ratio. A dihybrid cross is always this. Always, 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 always. That's a dihybrid cross. So that tells us everything we need to know, is that these guys are red, and that means if you have a big A and a big B, you're red, and that makes sense down here. These guys at the bottom are cream, so if you have 2 little A's and 2 little B's, you're cream. So that tells us what these parents are, because you have to have a big A and a big B to be red, because that's what we see here. And we know that these are true breeding. That's just a given for all these crosses. And so the parents look like this. So that's your typical example of true breeding stuff. Big A, big B, crossed a little a, little B, little B. That gives us this particular dihybrid in the middle. We get 9 to 3 to 3 to 1. Now, it always comes up, and it's always a good question, is how do we know that the 2 little B's goes with peach and the 2 little A's goes with orange? We don't necessarily know that, and it doesn't matter, because these letters are just sort of abstract representations. So you, as the experimenter, you could define these. You could say, you could change these 2 in the middle around, and that would be okay. All you have to do is make something that makes sense. So usually the way you'll see this is you'll be given this cross, and you'll need to assign genotypes to all of the individuals in the cross. So as soon as you see 9 to 3 to 3 to 1, you assign these genotypes to them. And if there are 2 that have a 3, it doesn't exactly matter. So orange could be on top here and peach could be on the bottom. It doesn't matter. All right, so that's one example, 9, 3, 3, 1. Now this chapter should be relatively easy because they're all the same. They're always going to be some modification of 9 to 3 to 3 to 1. Now, this case, there's not a modified ratio. It is an actual 9 to 3 to 3 to 1 ratio. So what makes this different than typical Mendelian genetics is that this is 2 genes involving the same trait. So before we had like height and color, here it's just color. So we have 2 genes that are involved in the color of the pepper. And they call this, the example is, they call it novel phenotypes. And what that means is we start out with 2 phenotypes, and if it was just like 1 gene, then we would get all red in the F1, and then in the F2 we'd get 3 red to 1 cream. And so that would be the same 2 phenotypes that we started with. But now we start to see new phenotypes come out. And because we see new phenotypes come out, it means, and it's a 9 to 3 to 3 to 1 ratio, that means there's 2 genes involved, and it also means independent assortment's involved, because the 9 to 3 to 3 to 1 is only true if independent assortment is involved. All right, so that's one example. Next example, the next example is they do a test cross for this. So, all right, that sounds cool. So let's take our red and cream, and we get all red. And we know that from the previous thing that these guys look like this, and these guys now look like this. So now we're going to test cross those. A test cross always looks like this. Well, a test cross, yes, so a test cross is always something that's homozygous recessive for both. In this case, that would be cream. And you always get a 1 to 1 to 1 to 1 ratio if there's independent assortment. And independent assortment is occurring in all of chapter 5. So, what are our options? We know that these are red. We know that these are cream. I don't remember what the other ones were. Where'd it go? Here's our red times cream example. For some reason, that didn't have a name. Let me give it a name. That should give it the name peppers. But anyways, there we go. Now it's called peppers. So let's see. So 2 little B's was peach. So 2 little B's was peach, and 2 little A's was orange. So a dihybrid cross always gives you 9 to 3 to 3 to 1, and a test cross always gives you 1 to 1 to 1 to 1. If we just did this cross, we really couldn't tell exactly what's going on with everything. We have to do the dihybrid cross first, but then once we do the dihybrid cross, we can predict what's happening in this kind of cross. The next example is a gene interaction. Then we start talking about epistasis. So the example they give us for this is, the first one is going to be recessive epistasis. Now, I'll just give you a little spoiler alert. In there, the ratio that is typical of recessive epistasis is going to be 9 to 3 to 4. So the example they use are labs. So these are dogs. If you take 2, and if you look at the book, you can see some cute pictures of dogs. And if you do this, you get all black, if they're true breeding in the first generation. And again, the best way to think about this is, if this is one gene, then we expect 1 to 2 to 1 and 3 to 1 in the next generation. But we don't see that. We see 9 to 3 to 4. And what we see is 9 black, 4 yellow. So those are the 2 things we started with, but we get 3 brown in the middle. Now we say, well, how do we explain this? Well, we think of 9 to 3 to 4 as modified 9 to 3 to 3 to 1. So every single time you do one of these examples, we'll do it. They use different letters in the. It doesn't matter. We can do it this way. It doesn't really matter what the letters are. You just have to be able to explain it. So again, a problem with this would be, you see this pattern, explain what the genotypes are of all of those things, right? And so if we do 9 to 3 to 3 to 1, we know that we have this general pattern. Now we just have to assign these different genotypes to the phenotypes over here. Well, the 9 goes with this. We can assign the 3 to that. And then here we have 4. So we're going to combine these 2 things, and those give us yellow. So what that means in this case, and the way I wrote it out in this case, if you get two little a's, no matter what you get at the B locus, you get yellow. So that means little a is epistatic to the B gene, we'll say. So that means if you're homozygous recessive or for the A gene, you get a particular phenotype no matter what's going on at the B gene. So that means that A kind of overrides B. So little a overrides B. And rather than say overrides, we say is epistatic to. So what that means is it says it's epistatic officially means stands on top of, or it sort of blocks it. But it only blocks it when it's homozygous. So that makes it recessive. Anytime that you have a situation where you only see a particular phenotype when a particular gene is homozygous, that means recessive. If you see a phenotype when a particular gene is heterozygous, that must be dominant. Okay, so that's it. So we've got this situation going on. So how do you explain this? So the idea here is that you have some kind of pigment that gets deposited in the fur of the dog, and it starts out yellow, and then it has to get converted to brown, and then it has to be converted to black. And we know that we have this kind of genotype where this is black, this is brown. I think I made this brown. And then these guys, if you have two little a's, no matter what's going on at the B locus, these are yellow. So what this means is that if you have two little a's, you're yellow. So that means the function of the big A gene, you need a big A gene to get from yellow to brown. If you get from, so this is like an enzymatic pathway. There's some enzyme that converts yellow pigment to brown pigment, and there's another enzyme that converts brown pigment to black pigment, and the enzymes are controlled by genes, and you need this A gene to be functional in order to go from yellow to brown. If it's not functional, then you get stuck at yellow. And the little a is a recessive allele that's not functional. So that's what that means. And then here you could have, if you have a big B, it goes to black. If you have two little b's, it goes to brown, or it stays at brown. But if you have two little a's, you never get to brown because it blocks it. And so that's what we mean by epistatic. Now, if you take yellow and so let's go up here. So yellow, we have a big A. Up here, if we have a big B, then it can go all the way to black. And so that's why these guys are black. But if this is little b, so that it gets blocked here, then it stays brown. So that's what we mean by epistatic. It takes two different steps or two different genes to get from yellow to brown to black. And if you block it at the first step, you're yellow. And if you block it at the first step, then it doesn't matter what happens at the second step. So that's what we mean by epistatic. All right, so another example and that's recessive epistasis. I don't think this is going to be another 15-minute one, but maybe we can keep it under half an hour. So this is another example of epistasis, in this case involving blood type. So we know that blood type, let's say we have two individuals that have some kids. And if they have a type A kid, that makes sense because we can get the A allele and the A allele. We could get an AB kid, makes sense. Get an A allele and a B allele. We could get type B blood because we got a B allele and a B allele. But there was a case discovered where these two individuals had a kid with type O blood. Now, if you were watching daytime TV, you might think that, oh, there's some kind of paternity issue going on, and everybody'd start yelling and screaming at each other. But it turns out that in this particular case, and sometimes that is the case, sometimes you can use blood type, kind of an old school way to determine paternity. But there's this phenomenon. This was originally discovered in India, so it's called the Bombay phenotype. And so, if you remember, blood type is due to putting antigens on the surface of a red blood cell. And you could have an A antigen, or we'll make it, instead of a color, we'll just use a different shape. Or you could have a B antigen. Well, those antigens are carbohydrates, which are added, that are made in the cell by a series of steps. And there's the first step, there's something called the H substance. And then that H substance can get converted into type A or type B. If you have the A allele, you get type A. If you have the B allele, you get type B. If you have the A and the B allele, you can get both. But there's some other thing here that has to first get converted to H. And so here we'd have the H substance here. And there'd be some other precursor here. So some other random thing down here. And so you have this H gene that needs to make the H substance. After you have the H substance, you can convert it into A or B. But if you don't have the H substance, then you don't get A or B because you're blocked in an earlier step. If you don't have A or B, then that means that you have type O. So that's where this came from. That means this person has HH. And then it doesn't matter what it has here. It could have AB, it could have AA, it could have BB. No matter what's going on here, if it's HH, it's going to be O. And that's because HH is epistatic to what's going on at the other locus. So let's look at the genotypes of this kind of thing. So what that means that in this cross, there's another gene involved. So we're used to just thinking about the blood type gene, but there's another gene involved. And so if you have this kind of thing, that's actually kind of a dihybrid cross. We could draw that out as a dihybrid cross, but it's a little bit weird because we've got this co-dominance going on. So we don't really have to, let's not think of that. But what we can think of is what are the probabilities? So what's the probability of getting different types of outcomes from this cross? Well, so this is something that is important. Whenever we're thinking about a dihybrid cross, we want to think about each individual gene separately. And if we think about just the H locus, so a dihybrid cross can also be thought of as two monohybrid crosses together. So we know that a monohybrid cross will give us this with the H locus. And in the case of the AB locus, we expect this. If we were just looking at this by itself. So we can say 2/4 this, so 1 to 2 to 1. So then we can calculate the probabilities of all the different types of blood. So we can, now if we get a big H, so if we get a big H, then blood type is determined by the other locus. So there's a 3/4 possibility of getting a big H. And if we get a big H and we get 2 A's, we multiply 3/4 times 1/4 and we get 3/16 is our probability of getting type A blood. So this is the phenotype. The probability of getting AB blood is 6/16. The probability of getting type B blood is 3/16. Because we have to get the big H and we have to get 2 B's. And those are the probabilities of those individual things. everything else is going to be, everything else is going to get this. And so 1/4 of everything is going to be type O, regardless of what you get with the A and B alleles. That's because type O is epistatic. Now this is an example because it's blood type and it's humans. We're not really, and we got codominance going on with the AB blood type. So it's not going to be 9 to 3 to 3 to 1. 9 to 3 to 3 to 1 is typical Mendelian stuff, where independent assortment is going on, and big A is dominant to little a, and big B is dominant to little b. But we're looking at modifications of that, right? So here we've got epistasis going on with our two little h's, but we've also got codominance going on with A and B. And so we're kind of combining those things together. But as long as we have independent assortment going on, then we can multiply these individual probabilities together to get those things. Now, it's a little bit sloppy, but this is 1/4. And over here we've got 6 plus 3 is 9 plus 3 is 12. So 12/16. So all of this adds up to 12/16, which is going to be 3. So 3/4 of the, so we've accounted for all possibilities. So 3/4 are going to have some kind of blood type, A or AB or B, and 1/4 are going to be type O. And so let's see. So here's what this looks like in the book. So if you look like this in the book, there's some kind of precursor. Here they call it an intermediate. That has to get converted to the H gene or the H compound. If you don't have the H compound, then you can't get to A or B. If you do have the H compound, then whether or not you have A, B, or O depends on what alleles you have at the ABO locus. That looks pretty complicated. It's not that bad. It's the same. So you have some kind of intermediate that has to get converted to H. If you convert it to H, then you can get A or B or O, depending on what's going on at the ABO locus. But if you don't have, if you're HH, so that means you have this HH genotype. If you're HH, you never get to this point, you never get to this point, and so that's always O. So we're getting blocked here. So that little h is recessively epistatic to what's going on at ABO. All right, so that's that. Now we're going to do 2 more types of epistasis. So that was kind of like a little side note on recessive epistasis, just another example of it. But the classic recessive epistasis is going to be the 9 to 3 to 4 in a dihybrid cross. So now we got another example in a squash. This is going to be dominant epistasis. And we're going to have some squash, and we're going to have 2 different colors. So these we're going to go by quick. White and green. And we get all white. All right, so let's go through the logic. White and green times green, all white. Well, maybe white's dominant, and it's 1 gene. If that were the case, then we would expect 3 to 1 ratio. We don't get a 3 to 1 ratio, we get a 12 to 3 to 1 ratio. And the 12 is white, the 3 is yellow, and the 1 is green. All right, so we interpret that as a modified 9 to 3 to 3 to 1 ratio. And what a 9 to 3 to 3 to 1 ratio tells us is that it's a dihybrid cross. So we know this. And a 9 to 3 to 3 to 1 ratio is always this. They use different letters in the book, but I like just using the A's and B's. And now we're going to combine these guys together. So it's always going to be 9, 3, 3, 1 with this pattern on the right. And then we combine them together in a way that makes sense to account for what we actually observe. So what this tells us is that in order to be green, you have to have 2 little a's and 2 little b's. So that makes this parent that. And in order to get this dihybrid, so what this is also telling us over here is if you have a big A, it's white, no matter what. So that's what we mean by dominant epistasis. So it's epistatic, meaning one thing is blocking the other thing. But in this case, it blocks it if we just get a big A. And it doesn't matter if we have a big A or a little a on the other allele. And so that's what dominant means. So like if we get this, it's white, no matter what's going on at B. So that's what we call dominant epistasis. That's where that is going. Okay. So that's dominant epistasis, because if we get a big A, it's white, no matter what's going on with B. So that means this guy has to have a big A. It could be big B here, or little b, or it could be anything at the B locus. But because we get this whole pattern where we get 9331, that means this has to be a dihybrid cross. And the dihybrid cross, and this has to be green. And if that's little a, little b, then the other one has to be big A, big B to fit the pattern of a typical dihybrid cross. And we won't worry about the biochemical pathway on this one, but it just gets combined together like that. And I'll show you in a minute, there's other ways to combine things in there, but they're all combinations of 9331. Now we're going to go over one more example, and then I'll show you that table that combines everything, and then hopefully this won't be super long. So the next one involves snails, I think. And you have albino snails, and you have pigmented snails. So albino, let's see. Can I draw a snail? Probably not. All right, so there's some kind of weird looking snail. That looks kind of like a snail. And if it's pigmented, it's pigmented. If it's not pigmented, it's albino. So the way this one, they start this one, you wouldn't have to start it this. I don't know why they start it this way. But this is the way they do it in the book, so we'll do it that way. So albino, cross to albino. Now, one thing I always point out, it's always, whenever I start writing albino, I always end up writing Albion because it's just muscle memory. But it's not an Albion cross to an Albion, it's an albino cross to an albino. When you cross these together, you get a pigmented. Now, so this is kind of a little bit weird the way they do this. So you get pigmented. So this is going to have a slightly different pattern as before. But then you get 9 to 7 pigmented to albino. 9 to 7. There's a 9. So that means what we're doing is all 9 look like that, and these get combined into the 7. This looks like this. And, sometimes I write these two in the middle a little differently. It doesn't really matter. So what this is saying is that in order to be pigmented, you need a big A and you need a big B. If you're albino, it's because you have either 2 little A's or 2 little B's or 2 little A's and 2 little B's. All of those give you albino. But 9 to 7, modified version of 9331, that means this is a dihybrid cross. If independent assortment is going on. So now we have to think about, so that means that pigmented have big A, so these are all the pigmented guys, and the albinos can be all these guys combined. So how do we know which albinos are up here in the parents? Well, we know that it has to be some genotype that gives us this, 2 dihybrids together. And it can't be big A, big B, because that would be pigmented. So the way it works is it's like this. Because we have to have, 2 true breeding albino snails mate and give us a pigmented. And because it gives us a modified 9331, we know it's a dihybrid cross. And so we know that these guys at the top have to have either 2 little A's, 2 little B's, or both. it couldn't be both because that would not, so we couldn't be, so we could do it like this. So that would be the way we've been doing it before, but then this would be albino. And then this would be pigmented. There's nothing wrong with that. And then we'd get this. I don't know why they didn't do it this way to begin with. And then these would be both pigmented. And then you'd get 9 to 7 pigmented to albino. So that would be fine. But in the example they gave, they said we started with 2 albinos. In order for 2 albinos to give us a dihybrid, they have to look like this. Because if we had 2 big A's and 2 big B's, well, it wouldn't be albino. And so we have, and if we had, they can't both, well, we'll come back to that later. But this is going to be an important thing. So 9 to 7, this is called duplicate recessive epistasis. And that's because little a is recessive to B, little b is recessive to A, and that's duplicate recessive epistasis. This is another case where, so we're going to have like 2 steps. So we're going to have some kind of precursor, some kind of intermediate, and then we're going to have some kind of pigment. So it's kind of like, it's exactly like the black, brown, and yellow, except, and so there's 2 steps. And let's say we need big A to get from this step to this step, and we need big B to get from this step to this step. So we need big A and big B to get all the way to the pigmented thing. If we have little a, we're blocked here. And we don't get to here. If we have a big A, we can get to this intermediate. But if we have 2 little b's, we get blocked here and we don't get all the way to the dark pigment. The thing that's different here is that these guys don't have, these are colorless, and these are colorless. So if they're colorless, that means albino. So your albino, if you're blocked at the first step, or if you're blocked at the second step, you have the same phenotype, and you need a big A and a big B to get all the way to the pigmented side. So that's very similar to the black, yellow, brown thing. It's just that the precursor and the intermediate had different colors in that example. So that's where we got 934. So 9 to 7, modified Mendelian ratio. And it's called duplicate recessive epistasis. And then here's the table. So this is table 5.2 from the book. And 9331 this is like no interaction really. So that's just like your typical Mendelian ratio. But you could also get the novel phenotype thing. 934 is recessive epistasis. 1231 is dominant epistasis. 9 to 7 is duplicate recessive epistasis. And those are the examples that we just went over. And then there's some other examples, but we're not going to do those. So, but 961, 15.1, 13.3, those are, you can get all kinds of different gene interactions. But the key here is focus on these and know that they're all modified 9331 ratios. Here's those abbreviated genotypes. All right, so that's the end of that one. So hopefully that wasn't too long. I'm not doing a very good of timing them.